Assalamualaikum and greeting for examiner. My name is Muhamad Alfishah bin Rizal. My matric number is 72378. The title of my proposal is Enzymatic Hydrolysis of Selected Starch for Fermentable Sugar Production. And this proposal was supervised by Dr. Dayang Salwani, binti Awang Adeni. This presentation will be covered introduction, literature review, military and method, expected outcome, work schedule, and references. And then we will proceed to the introduction, which is research background. Starch is the most prevalent carbohydrate in the human diet, and it may be found in almost every plant food. Other starch-producing plants such as tapioca and wheat, sago, plant, potato, cassava, and rice among the recognized plants with high starch content, which is a natural resource. Now we go to problem statement. First is enzymatic hydrolysis of sago starch, corn starch, tapioca starch to glucose must be enhanced in order to utilize the selected starch for lactate and ethanol fermentation. Next, sago starch content is a key element impacting the efficiency of the enzymatic hydrolysis in this project. As a result, these research objectives are to produce fermentable sugar from corn starch, sago starch, and tapioca starch using commercial amylolytic enzymes, and to determine the total of fermentable sugars production from 3 different sources upon completing enzymatic hydrolysis process. Alright. Now we proceed to literature review, which is characterization of cassava starch. Cassava starch can be converted to glucose in 2 ways, which is enzymatic hydrolysis and acid hydrolysis. Last but not least, many industries benefit from cassava starch outstanding properties such as high paste viscosity, high paste clarity, and high freeze-store stability. Now we go to characterization of corn starch. Starch is a renewable polysaccharide commonly used in the food packaging area due to its abundant, low cost, and commercial availability. Native corn starch easily gelatinized, and it is capable of forming edible films EFS by thermoplastic processing, but low shear resistance, thermal decomposition, and high tendency to exhibit retrogradation limit its use in some industrial food applications. EFS based on modified cornstarch are clear, flexible, and provide selective barrier to carbon dioxide and oxygen, however show a poor water vapor barrier. Then we go to characterization of sago starch. Sago starch has a multitude of uses in both food and non-food applications. In food processing, sago starch has been used as a stabilizer and thickener. The textural properties of the gelatinized and retrograded starches are important for their potential use in food system and depend on the molecular component and structure of the starches, which is amylose and amylopectin. Last but not least, amylose leads to higher viscosity values and more pronounced shear thinning behavior. Alright, let's proceed to starch for fermentable sugar production. A natural conversion of simple carbon sources such as sugars and amino acids by yeast to produce alcohol is known as fermentation for alcohol production. Next, starch is converted to glucose through 2 processes, which is liquefaction and saccharification. Therefore, starch is broken down into soluble dextrin and oligosaccharides in a short period of time. Now we go to theory of starch to glucose conversion. The hydrolysis of sago starch to examine the impact of various variables such as starch or enzyme concentration, pH, and different types of starch. Next, liquefaction required thermomil 120L, while salicylification required dextrozyme. Therefore, increasing the hydrolysis volume led to a longer incubation period for saccharification. Alright, last point of literature review, which is bioethanol. To make bioethanol, organic material is fermented into simpler chemicals like sugars by microorganisms in the presence of oxygen. Microorganisms subsequently convert these chemicals into ethanol and carbon dioxide through the process of fermentation. Therefore, ethanol synthesis has been attributed to a wide range of microorganisms like bacteria, yeast, and fungal strains. Alright, now we proceed to materials. There are 5 materials in this project, which are cornstarch, sago starch, tapioca starch, hydrolytic enzymes, and dinitrosalicylic acid DNS reagent. Next, we go to method. There are 5 methods in this project, which are preparation of cornstarch, sago starch, and tapioca starch slurries, liquefaction, saccharification, analytical methods, and experimental methods. For the first step, which is preparation of cornstarch, sago starch, and tapioca starch slurries, is to produce the starch slurries for 10%, 20%, and 30%. The second step is liquefaction. In this liquefaction method, the starch slurry will be mixed together with thermomil 120L. Then, the third step is saccharification. In this saccharification, amyloglucosidase will be added to thermomil treated liquefied suspension. The fourth step is analytical method. There are 2 tests in analytical method, which are iodine test colorimetric method and dinitrosalicylic acid DNS method. The last method is experimental method. In this experimental method, it is to produce the starch concentration of 10%, 20%, 30% to determine the effect of starch concentration in enzymatic hydrolysis of selected starch. Alright, now let's proceed to expected outcome. It is expected to produce the highest fermentable sugar from selected starch, and it is also expected that different amount of total fermentable sugar will be produced from different types of starch. Alright, now go to work schedule. This is my work schedule for this proposal. It starts from September 2021 until May 2022. Last but not least, this is the references for this proposal. Thank you.